All right. Welcome, everyone, to the SLAM seminar by Dr. Mehrana Nejad from Harvard University. Mehrana got her PhD working with Julia Yeomans at Oxford, where she was supported by the Clarendon Scholarship. She, for her PhD work, was a winner of the thesis prize from the Institute of Physics, the Theory of Condensed Matter PhD prize. And she worked on a number of topics in the Yeomans group, which informed her branching out into her current research with the Mahadevan group at Harvard, working on active nematics, working on order and soft matter topics in living tissues. And I think we're going to hear about some of those themes today and some of her more recent work. So take it away, Mehrana.
Thank you. Thank you for the kind introduction. Hi, everyone. Today I'm going to show you some of my recent research about geometry and shape in thin living layers. Please feel free to ask your questions. I think you can, and I'm happy to answer them while I'm talking. I'm going to talk about my recent research on shape and geometry of living systems, in particular thin living layers. These are suspensions, and I will show two examples. The first one is thin active nematic layers, and the second one is a seaweed which deforms and changes shape as it grows. Okay. So here, these are some examples of systems I'm thinking about. The first one is a suspension of active filaments. These are microtubules, the filaments that we all have in our cells. And as you can see, they form these interesting patterns that I will talk about in more detail soon. The second one is a bacterial droplet on a substrate, and the way it invades its surrounding with fractal type patterns at the interface. And the final example is the seaweed I'm going to talk about. This is one of the largest single cells, and as you can see, the scale bar is large, so it's a centimeter, but it's still a single unit, it's a single cell. And so there are a diverse set of questions we can ask in all of these systems. For example, what are the instabilities and patterns that form, how they form and how they evolve? And what sets the spatial and temporal correlations in these patterns? Sorry, I don't know why it goes back. And how does the morphology and geometry of these things change over time? And what are the range of shapes and morphologies that we can get in these systems, constraints from mathematics? And also, for all of these questions, we can look more broadly and ask all of these questions in an evolutionary time scale and look at how the morphology changes during evolution. For example, here I have an example of the seaweed I showed you earlier over the course of long time, the evolutionary time. And you can see that the morphology changes a lot during this time scale. Just a brief introduction about the first part of the talk, which is going to be about active nematics. This is a bacterial system. It's a bacterial suspension, and the individual units, each of them is a bacterium. So if we pick a unit in this system, the shape and geometry of this unit has a shape of an ellipse. So it's like a two-headed arrow. We call it because it has a nematic symmetry in the shape. But also because these systems live in a low Reynolds number regime, which is the regime where the inertia the momentum, the acceleration is small. So the first order active force that we can consider this system with is a force dipole. So the force that they exert on their surrounding also has a nematic symmetry. And to be able to look at the dynamics of this system over time, we need to define an order parameter which conserves this symmetry. So this, which accounts for this symmetry. And this order parameter that I show by Q is called a nematic order parameter, and it's a rank 2 tensor. And also to explain the dynamics and to understand the dynamics, we need a velocity field, because these units, they change their orientation. And we know that for the flow field, we can always use the Navier-Stokes equation. The difference with the passive non-living systems is that here we have the activity because each of the units exerts a force on the fluid environment. So we have an extra active stress in these systems. And I've shown the active stress here is proportional to the nematic tensor. It's along the nematic tensor because the units, they exert the forces along their shape, the elongated shape, and there is a coefficient here. And this coefficient, it could be positive or negative, and depending on these details, these systems are called contractile and extensile. And then, of course, to look at the dynamics, we need to look at the orientation, how the orientation of these units change over time, which means how this rank 2 tensor changes. And the way these tensor change is due to the active flows that I've shown by S here. So the different parts of the flow lead to deformation and reorientation of these systems. For example, the gradient of the flow field, which is the strain rate, that leads to, because the units, they don't have a spherical shape, they have an elongated shape, they rotate on the strain rate, and the strain rate can align them. And the second part of the flow, which is the asymmetric part of the velocity gradient, is called the vorticity, and that also rotates the units independent on shape. And in addition, because these systems, the units are in contact with each other, the elasticity also leads to reorientation and change in the dynamics of the orientation. In addition, the system is confluent. It's the units, they interact and they align and terms related to that in this equation. And then basically the
Mehrana?
last, yes?
Sorry to interrupt. For many in the audience, the connection seems to be, from your end, seems to be a little jumpy. I'm wondering if temporarily turning off your video would help us at least hear your audio more smoothly.
Oh, I see. So let me. Is it better now? I hope so.
So far, we'll find out.
Mm-hmm. Okay. Um, Sorry. So, okay, just to quickly summarize, I don't know why the slides are jumping back. So we have a set of equations, which are the equations for the velocity field generated by the units, and we have set of equations for the orientation of these units. And these two equations are coupled because the units produce the flow and the flow reorients the units. And then if we solve these equations, either do linear stability analysis or solve them numerically, we find that due to the active flows, any small perturbation in the orientation field grows over time and leads to formation of interesting orientational patterns in these systems. For example, here is an example of bacterial suspension. you see formation of defects. These interesting structures, they are called defects. And in a system with a nematic symmetry, the most important defects are plus half and minus half defects. And these are shown here. And as you can see, the has a polar shape. And because of this shape, any active flow leads to motion self-propulsion of this defect. Whereas minus half defect, it has an isotropic shape, and the flows, they cannot move the defect. And a very important concept here is that these defects, they cannot be formed or annihilated in a single, each defect cannot be formed or annihilated by itself. You always need to have two of them to see defect formation and annihilation. And so these defects are called topological. And so there are lots of research about how these defects interact, what are the orientation of the defects respect to each other, what defects form in 3D. For example, in 3D, it's very interesting because these defects, instead of point-like things, they form discrimination lines, which are 3D structures. And okay, so if we go back to the movie I showed earlier, we can now distinguish these defects. And we see that the plus half defects, they move around, and the minus half defects, which are shown in blue here, they don't move that much. And the little bit of the movement that you can see is due to the advection by the general flow. It's not a self-propulsion. And so I mentioned that the defects are topological, and this has a very interesting consequence for biology. For example, here you can see Hydra. It's a marine creature. And in experiments, people have seen that this creature forms arms at the position of defects. And since these defects are topological, this means that there is a constraint on the number and position of the arms that this creature could form. And so the number of arms cannot be easily changed because it comes from a topology constraint. And so this is how physics or physics concepts could play an interesting role in biology. Okay, so the systems I talked so far, they were mostly 2D, and we forget about the thickness of these systems. We assume that we have a completely 2D system without any changes in the thickness or in the shape. But there are examples where, especially in biology, there are many examples where the thickness of the layer is important. So I'm going to show you one of these examples, which is the seaweed I've shown here later in this talk. But there is also many other cases. For example, epithelial cells. These cells, they deform, but they also change their thickness. And this is very important during development. For example, in embryo, in different animals, gastrulation, which is an event where different organs form in these systems, involves thickness changes and deformation of thin layers. And so a purely 2D theory, or a theory which doesn't account for the thickness change, is not able to understand these developmental events. Okay, so these are the examples I briefly mentioned about why the thickness matters, but now I'm going to quickly and briefly show you how one could account for the thickness changes in these systems. So let's assume we have a thin layer which is deformable, so you can have an optional geometry. It has an optional geometry, as you can see here on top, and the thickness also varies in a space. And there are works on thin films that change their shape. But all the previous studies, they have ignored the thickness. So they assume that the thickness is fixed and that there is a 2D world, meaning that the thin layer is not embedded in 3D. So the world only consists of this film. Whereas we know that in biology, the thin layers, they interact with the surrounding, and it's important to consider the embedding of these layers in 3D. So the goal of the research I did here is to look at the thickness changes and consider the thickness changes, and also consider the embedding of this film into the 3D environment. Okay, so to do that, to simplify the math, the best way we could do this is to define a coordinate system which aligns with the direction of the curvature lines. And so the coordinate system that I've shown by E1, E2, and N consists of two coordinates which are aligned the curvature lines of this film. And the third axis is perpendicular to the film. And we can always define the position of the center surface of the film. And we put the coordinate there because that simplifies the equations. And then having defined this coordinate, we can easily find the metric tensor using the position of the center surface and also the external curvature. And we need this external curvature because we need to account for the embedding of the film in 3D, because we're interested in looking at how the external world could change the dynamics of the film. And then we can look at the deformation of the film by defining the dynamics of this center line. And this gives us the velocities in three different directions. Two directions inside the film and two directions perpendicular to the film. But note that this velocity is the velocity of the center line of the film and not the velocity of the fluid inside the film. So we have three different velocities here. Two different velocities here. One is the velocity of the center line, which shows us how the deformation evolves in time. And the other is the velocity of the fluid inside this thin layer. And the velocity of the fluid in the thin layer is given here by the Stokes equation. And now, because the curvature, the coordinate system is evolving over time, we need to account for the covariant and contravariant components of the tensors and the velocities. So these are very similar equations as the ones I showed earlier. The only difference is that now I've accounted for the coordinate system, which is evolving in time, and it's a curvilinear coordinate system. And now to look at the dynamics of this nematic tensor, we have to be careful because the coordinate is moving and deforming in time. So this adds extra terms in the dynamics.
Oh, can I ask that you don't have a
Yeah.
separate density variable? So is the layer the region of space where the Q tensor is above a threshold, like in allotropic theory?
Okay, so you mean, yeah, that's a really good question. I assume that the nematic is confined inside the film and outside it's a fluid. And the way it works is by a boundary condition that I'm going to show
Okay,
now.
okay.
Yeah. So yes, so to complete these equations, we need to account for the boundary condition, which is which for this study I considered a traction-free boundary condition, meaning that there is no external force on this thin layer. This can be easily generalized for the cases where, for example, there is a surface tension or surface tension gradient on the top and the bottom of the film. And the other boundary condition is the boundary condition associated with if the material is able to go outside the film or leave the film or not. And this is called the kinematic boundary condition, which I've shown it here, and it's in the curvilinear coordinate system, which is why it's the form is not is quite different from the case where the coordinate is fixed. So yes, so here I don't allow the particles to leave the interface, and that's given by this boundary condition. Was that your question?
Yes, that's what I was
Yes.
wondering. Yes,
Okay, yeah.
thanks.
And so now that we have the boundary conditions on the top and the bottom, and we have the dynamics of the nematic tensor, the orientation of the units, and the velocity field, we have everything. But as you can see, this is very hard. This problem is very hard to solve analytically, but also numerically, because numerically it's hard because there are two different length scales in this system. One is the thickness and the other is the length, and these two different length scales are very different. So it's not easy to solve these equations numerically. As a result, it's good to do a lubrication approximation, which means if we define the aspect ratio of the film, we know that it's small, and it can help us simplify the equations and build an effective theory for this system. Okay, so this epsilon parameter is the aspect ratio of the system. And now we can expand all the fields in this problem as equations order by order. For example, here the length scales along x1, x2, which are the two directions inside the film and perpendicular to the film, scale with epsilon as I've shown here. The x1 and x2, they don't have epsilon because they're along the long axis, but across the thickness, because the thickness is small, the length across the thickness is proportional to epsilon to the first order. And also we need to make the assumption about the curvature. If the curvature, how the curvature scales with the aspect ratio. For example, if you're thinking of a thin flat layer, then the curvature to the first order is 0. And so the curvature is proportional to epsilon. Whereas if you're thinking of a film which is curved, for example, in many cases like Drosophila, the curvature is not negligible. And in that case, to the first order, there is a constant curvature. So the curvature is not proportional to the epsilon anymore. And this, the assumption, especially about the curvature, would change the final equations that we get after doing the mathematics, following the steps. Then we need to expand everything as a function of epsilon, like all the velocities and all the other fields. And when we expand everything, we can just solve the equations order by order, account for the boundary conditions, integrate the equations across the thickness, and build an effective theory for this thin layer. And so here I only show the results. For example, in the case where we completely ignore the thickness, the traditional active nematic theories, they found that if you look at the instability in the nematic phase, you see the dispersion relation looks like the black curve I've shown here. Whereas when I do the theory and find the effective equations, I find that this instability changes a lot when you consider the thickness. And to have an intuitive idea why this happens, it's contractile case, contractile activity leads to thickness variations. And the thickness variations in many cases appears before the variations before the instability in the nematic. And as a result, the dispersion relation changes a lot when you account for the thickness. In the curved case, for example, now that when I build the equations, you can just put in whatever geometry you're interested in. And this you can do that through the curvature tensor. So if I consider the curvature tensor to be given by the cylinder, then I can look at the instabilities that this active nematic thin layer on a cylinder forms. For example, here are two examples for the contractile and extensile systems. In the contractile case, the instability happens in an isotropic phase. The instability happens by the formation of bands of nematic order along the long axis of the cylinder. In the extensile case, it's the opposite. The nematic order forms along the circumferential direction. And one of the interesting predictions of this theory is that when you account for the thickness and the curvature, the shape and the thickness are coupled. So an instability in the shape could lead to an instability in the thickness, and an instability in the thickness could change and deform the shape. And so, yeah, this has many applications. For example, one thing I've been working on is, in collaboration with the experimentalists, is to understand evaporation of active droplets. So here you have substrate, and it spreads and forms a thin layer. It has microtubules and motors in it. And we found that it forms very interesting patterns and self-organization patterns in experiments. And this problem in general is very hard. Even without activity, it's really hard to understand the dynamics of this droplet because of the presence of many inclusions, for example, polymers, motor proteins, and everything. But so I had to do lots of assumptions, and this is not at all a very complete theory, but at least we understand some of the features. For example, one thing that they can look at in the experiment is to look at the dynamics of the contact line. So this is a movie from the experiment. When they start the experiment, the droplet first shrinks, and that's firstly because of the evaporation, but also due to the activity. And the interesting new behavior that happens here is that after a while, the droplet starts expanding again. And this is only because of the activity. And in the passive system, you can't really see these fast dynamics at all. And the reversal activity plays an important role in it. I'm not going to go to the details about this because I'm going to talk about the other system, but this, I just wanted to show you one of the applications of such a theory.
Okay, can I ask about the
Mhm.
the compressibility? Usually, incompressible Navier Stokes is assumed in active nematic theories, but I'm guessing that can't work here, right?
Exactly. Yeah, that's a really good question. Let me go back to, yeah, the way you're talking about the evaporating droplet, right?
Right, but more generally in this construction too.
In this construction, I do have the
You do have it.
incompressibility. Yeah. And the incompressibility plays an important role, and the role is it gives you the change in the thickness. Because I consider it to be incompressible when there are in plane flows that look like compressible. They're not compressible because there is a third component perpendicular to the film, and that
Mhm.
leads to the thickness changes.
And is that still true then in the evaporating droplet
In
case?
the evaporating droplet, no, it's not true because you're losing material. And the
Right.
way, and yeah, exactly. And the way it works is that in this boundary condition, the kinematic boundary condition, which is the boundary condition that enforces the material to stay inside the film, you need to have an extra term due to the evaporation because you're losing the material.
Great.
Um,
Thank you.
and yes, exactly. You still have incompressibility in the bulk, but at the interface, you're losing the material, which is why we have to change the boundary condition on the top, where the droplet is in contact with the air. But that's a really good point. Yes, that's exactly one of the important things about this system. Um, and yes, and the second example is this seaweed that I'm going to talk a bit about. Uh, okay, so this is a work in collaboration with Meyerowitz's lab in Caltech. There are three experimentalists that I'm working with. They have this fascinating system, which is called Caulerpa. It's a seaweed, and it has this interesting dynamics and geometry. The thing that for me was very interesting about this is that the entire thing that you see on the left, it's a single cell. So like this is huge. It's and this is like the scale here is centimeter. And because it's a single cell, we can look at the dynamics using the same framework I introduced earlier. And so here in this movie, the propagation of the green stuff that you can see is the chloroplast. And the dynamics in general is coupled with the daylight dynamics. And so it's oscillating due to the day-night change in the sun and the illumination. And in the experiments, they can change these conditions. For example, they can change the period of the illumination and look at the growth and the shape over time. Okay, so to understand the shape and geometry of this system, I consider this coordinate system where I define the tip and I look at the distance from the tip in the edge. I call it S. And there's another direction perpendicular to the rim, and show that by N. So to understand the geometry better, this leaf-like thing is like a 2D system. It's 3D, but it has a small thickness, but it also has a cell wall. So inside, instead of a membrane that animal cells have, this one has a cell wall like many other plants. And the cell wall is a thin layer of fluid, and so it deforms, and that's what I'm going to look at. So that's the thin layer I'm thinking about. And so yeah, we can define these coordinates. Plant, there is a thickness along the Z direction in the plant. There is a thickness associated with the cell wall, which is like the membrane of this system. And the way it grows for this specific system, we don't exactly know, but in many other cases, it's known that the way that the system grows and changes shape is by the active filaments inside the cytoskeleton of this system, which move the vesicles, which are the material necessary for the growth, to the tip of the cell. And then they merge the vesicles to the cell wall, and then the osmotic pressure, which is the pressure difference inside and outside the leaf, leads to the growth. Because when these materials join the cell wall, the pressure leads to the deformation, because these are fresh material and they haven't solidified yet. And so, yeah, the goal is to understand the morphology and shape of this plant, this seaweed, as a function of the deposition rate of the vesicles to the cell wall, and as a function of external properties of the system. For example, you can change the pressure, or you can change the concentration of salt inside the fluid that the seaweed lives in. And in the experiments, they can put gold particles in the cell wall and look at how they move, and that gives us a feeling of the strain rate in the plant. And for example, here in this movie, I try to show one of these experiments where they track the gold particles and they look at how they deform and where they move to. So in this theory, the thin layer that I'm considering is the cell wall, and it's not the thickness of the leaf. And so, yeah, the goal is to link the mechanical properties of the cell wall to the shape and morphology. And to do that, we account for the fact that the viscosity of the cell wall changes as a function of the distance from the tip. And the deposition rate of the vesicles also change as a function of the distance from the tip. because when the vesicles join the cell wall at the tip by the microtubules or other filaments, they're fresh, they're new, so they have a lower viscosity. And so that changes the material property as a function of the distance from the wall. Time passes, they solidify, they undergo some reactions and chemical interactions, and so the viscosity increases as a function of time, which means the viscosity increases as a function of the distance from the tip. So we have to choose two functionalities, one for the viscosity of the cell wall and the other for the deposition rate. And then we need to, what we are going to find is the shape of the leaf, which includes how the curvature changes across the rim. It also includes how the thickness of the leaflet changes as a distance from the tip, and also how the wall thickness changes. And so repeating the procedure that I talked about earlier, we can find an effective 2D description, which is the effective stress tensor along two different directions, and the effective force balance along the rim, and the effective force balance perpendicular to the rim. And the effective force balance perpendicular to the rim has a pressure term in it. It's an osmotic pressure, and it's a pressure difference inside the plant and outside. And it's because it has organelles and nuclei inside it, so there is a bigger pressure inside. And whenever the wall is fluid, the viscosity is low, this pressure difference is able to deform the shape. And that's where this term comes from. And we have another equation for the dynamics of the thickness as a function of the distance from the tip, and also as a function of time. And that has a term due to the velocity of the fluid inside the wall, and it has a term because of the deposition of the vesicles to the cell wall at the tip. And so this set of coupled equation, then these can give us the morphology and shape as a function of time.
Is elasticity of the wall playing any role?
The viscosity?
Elasticity.
Oh, elasticity. Okay, yeah, that's a good question. We assume that it's completely viscous, so we ignore any kind of elasticity. But you're right that the elasticity at a small time scale could play roles, but we ignore that for now. And we assume that the viscosity goes to infinity at very large distances from the tip, meaning that the thing is completely solidified.
Okay.
But at a small time scales, yeah, the elasticity plays a role, especially when they deform, because in the experiments they're also able to deform and pull the plant and see how that reacts to this. And in a small time scale, it does show elastic behavior. But we only look at, because the growth rate is pretty small, it only grows maybe half a centimeter a day. So because of the because of the long time scale of the growth, we assume that it's the growth could be explained only by viscosity and not by the elastic properties. Okay, then we can solve these equations considering the symmetries and find the solutions of these equations. And then we get a diagram that shows the morphology of the tip for different values of the parameters. For example, for the length scale over which the deposition of the vesicles to the cell wall happen, and for the length scale over which the wall solidifies as a distance from the tip. And what we found is that depending on these two length scales, we can see two different morphologies. One is where the tip is convex, and the other is when the tip is concave. So we can go from positive curvature of the tip to negative curvature by changing these two length scales, but also changing the pressure difference inside and outside. And in the experiments, they're able to modify the conditions to account for these sorts of perturbations or changes. For example, they can add salt or other things to the solution, and that changes the pressure difference inside and outside. And as a result of that, they can also see a change from a positive curvature of the tip to a negative curvature of the tip. But it's very hard to do any quantitative comparison between the model and experiment. It hasn't been very easy so far, because if you want to change the material properties of the cell wall, it normally leads to other kind of things that one does not normally predict. For example, it changes the biology, and there are feedback loops, and it changes the growth as well. So the only way we have been able to compare the theory and experiment so far is by changing the pressure outside the plant by adding salt. And even that, if the change is very big, the plant is going to stop growing. So there are certain regimes that we can only compare the theory and the experiments. But at least it's promising to see that you can change the pressure outside and see the same sort of morphology changes, curvature going from positive to negative, by changing the external properties of the fluid. And so yes, here is a summary. In many cases, I showed that the 2D active nematic theories are good. They're able to understand spatial and temporal patterns in living systems. However, there are many cases where the thickness is important and the deformation of the thin layer is important. And in these cases, especially in contractile systems, one needs to account for the thickness. If one is interested in more biological developmental problems, then the thickness plays an important role. And in many cases, the thickness instabilities appear before the instabilities in the orientation field. And I also showed some examples of how these theories can be helpful in understanding morphology and geometry of living systems in a seaweed, Caulerpa, and also in evaporating droplets. And yeah, thank you for your attention, I think.
Right. Thank you, Mehrana, for a wonderful talk. So floor is now open for even more open for questions. So please feel free to unmute and ask your question.
Hi, I can go first. So, thanks for the presentation. And like my first question was, so we have some boundary conditions for the Q tensor, but just in terms of like alignment, are we specifying alignment near the boundaries in the equations, or like what set of boundary condition is there on Q tensor?
Yeah, no, that's a really good question. For this, for the problem I showed you, I consider the case where the nematic is aligned with the film. So this is like a constraint that I put on the theory, meaning that the orientation tensor does not have any component perpendicular to the film. So it's completely aligned within the film. And I assume that the activity is constant across the thickness.
Got it. So like tangential
So
boundary condition.
yeah, condition is not, the nematic has to stay inside the plane, but it can rotate inside the plane and form different structures and patterns.
Got it. And that also seems to be like a like an not approximation, a convention that we follow in the literature, that we place it as like tangential boundary condition, or no normal bound component. But do we see that in experiments as well with your leaves? and um,
Mm-hmm.
like evaporating cells?
Mm-hmm. Yes, yeah, I mean it's a very diverse. There are diverse cases where we can use this theory to study, but there are limitations as well. So as you mentioned, in many cases, the force might be even perpendicular to the interface. It may not be inside the film. For example, in epithelial cells during gastrulation, it happens that the force is perpendicular to the film instead of being inside the film. So that's another regime which is interesting to study. But in the cases that I talked about, the evaporating droplet, the filaments, they are parallel to the interface and to the surface, the substrate. The theory for that is a bit different from the theory I showed, because now there is a substrate, whereas I didn't really talk about the substrate. I assumed a film in the earlier example that I showed. So that theory has another boundary condition because of the presence of the substrate. And that could be anything. Like in this case, we found that probably the free slip boundary condition is good, but in many cases it's no slip for the velocity. For the nematic, it is parallel orientation because we can see the orientation of the filaments in the experiments and they're parallel with the substrate. For the plant, we don't really have the nematic. It's an isotropic fluid. It's active because it grows, but it's we don't have the nematic, so we don't need to be worried about the orientation field there. But the orientation field is important and it could change things. If it's perpendicular, it would change things.
Yes, thanks a lot. That
Thank you.
answers the question.
Henry has a question, has his hand raised.
Yes, thanks a lot for the talk. I found it very curious that you get this transition from convex to concave in the tip of the leaf. So I was wondering whether in particular where it's concave, this cusp is forming, do you expect some sort of universal shape that this cusp should have? Because there, I guess it's very simple, like what's going on, you can linearize just around the tip itself.
Mm-hmm. No, it is very interesting, and I'm very interested in looking at the cusps and things. However, here we didn't really look at cusps. I think universality and looking at the shapes of a cusp in active systems is very interesting, and there are lots of interesting biological applications for that. But in this case, it's when the curvature is negative, it doesn't really form a cusp. It's a smooth negative curvature in the experiment, and in the theory, I also looked at the smooth cases. I didn't look at the singularity solutions.
Yeah, but I guess it might still have a functional form that's somewhat generic.
What do you mean? Because for the cusps, yes, you can always do that. For the non-cusps, if there's no singularity in the curvature.
Is there a certain maximum inward curvature that's set by some like scale in the problem?
Mm-hmm. Yes, exactly. That's set by length scale related to the deposition rate and the length scale of the thickness, the distance between the top and the bottom planes. So like a combination of these two play a role in the length scale of the morphology.
Okay. And it becomes then an exact cusp when you would take the limit of infinite thin cell wall?
In the infinite cell wall, you can't get this morphology. If it's a shell instead of a fluid, meaning that the cell wall has a fixed thickness, you can't see this morphology.
Okay.
Because the negative curvature, it comes from the combination of the cell wall changing its thickness from the tip And also the leaflet changing its thickness from the tip. So like the distance between the two planes changes from the tip, and also the cell wall thickness changes from the tip. So yeah, the thickness is very important in the negative curvature morphology. And we do see that in the experiment as well, that the cell
That it
wall,
becomes thicker.
yeah, the cell wall thickness changes.
Great, thanks a lot. That's super interesting.
Thank you for your question.
Okay, it looks like we have another question from Gangcham.
Hi, so me again. But I just wanted to like clarify. So the curvilinear or like the curvilinear geometry in the problem comes from the fact that we're approximating the system to be two-dimensional by the aspect ratio being very much skewed. Right. But let's say in theory we just completely solve the Q-tensor equations in 3D, then it would still be Cartesian coordinates like in a regular 3 dimensions, right?
The choice of the coordinate system is just to make things, because when you choose this coordinate system along the curvature lines, many of the terms become zero. The curvature tensor becomes diagonal, which means you lose some of the components, and that simplifies the equations a lot.
Yes, so that is true. But let's say I'm just solving the Q-tensor in 3D, like in the bulk of the system. Um, it would just be a different system with curved boundaries instead of uh, like my system being in curvilinear coordinates.
Yes, you can use the normal coordinate, but then you have to account for the bound, because it's a moving boundary problem. The interface is also evolving over time. So if you solve for the Q tensor, you have to solve for the dynamics of the interface as well.
True. And that coupling is very much difficult, I assume.
It is very difficult, especially because if you want to do simulations on long length scales, because the thickness and the length extension is very different. These are really heavy.
I understand. Yes, thanks.
So I was wondering, toward the end of the first part of your talk, you showed these very cool different morphologies of deformed tube we could get. And so what problems in morphogenesis do you think that'll be most applicable to? And do you think you could apply this framework to the Hydra and get some useful information?
Yes, I'm mostly interested in looking at how gas relation works in different animals, because they have different, the shape of the embryo is very different in different animals. And so how the shape of the embryo, which is the geometry of this thing, would change the way gas relation happens. And the gas relation, if anyone doesn't know about it, is just when you have the embryo, when the different organs form, gas relation happens, which means the embryo folds inside and it changes its thickness, and that leads to formation of different organs. And because the embryo has different geometries in different systems, this is like, for example, one of the cases I'm interested in looking at. Unfortunately, there are not many experimental data on the 3D experiments on embryo gas relation, but yes, this is a direction I'm thinking about.
Okay, any further questions? I'm going to stop the video here.